today. I'm a member of LifeNet Health's Life Sciences team, and I'm excited to be hosting this session. Before I hand the mic over to our speaker, Dr. Ed LeCluyse, I'd like to take a minute to go over a few housekeeping items about this webinar. First, if you're experiencing any issues during this webinar, please text Stacy at 757-298-3183 for technical assistance. Next, today's webinar will be available on demand soon after the live session, and we will send you instructions on how to access the recording. Lastly, we'd love to hear from you during today's presentation. If you have a question for our speaker, feel free to send it through the chat icon directly at the bottom of the WebEx interface. Please send your questions via chat directly to Stacy Hansen, who will collate them for us. We'll be answering questions at the end of the session. If we don't get to your question during today's webinar, we will be sure to follow up afterwards. Now, let me start off with a brief background on LifeNet Health, and then Ed will take over from there. LifeNet Health is a nonprofit organization that's been in business for over 35 years and has 1,200 dedicated team members. We're based in Virginia, but we have locations across the world. LifeNet Health is a mission-driven organization focused on improving patient lives. We are organized in 3 distinct centers of excellence. We are an organ procurement organization for Virginia, facilitating life-saving organ transplants. We provide human tissue allografts. We process human tissues for clinical use for surgical implantation in sports medicine, spine, trauma, wound management, dental, and cardiovascular applications. Lastly, Life Sciences. This is a newer division focused on leveraging LifeNet Health's vertically integrated infrastructure to supply the highest quality of human tissues, cells, and models for preclinical research. Saving lives, restoring health, and giving hope isn't just our mission. It's been our sole motivation since the 1980s. That commitment has driven us to become a world leader in allograft bioimplants with an unmatched safety record that makes us the most trusted partner in the field. It also has focused our efforts to aggressively innovate bioimplant technologies and to advance preclinical research by providing access to human tissue, cells, and cellular models. We work very closely with our donor families, and we're determined to honor the gift that their loved ones have unselfishly donated in order to save lives, restore health, and give hope. In 2019, we facilitated 585 organ transplants. That's 585 patients that were on the organ transplant wait list that received the gift of life. Our clinical tissue allograft franchises delivered over 700,000 tissue allografts for surgical implantation. Our extensive network and infrastructure enable us to work closely with 8,000 donor families to assist in donation. LifeNet Health Life Sciences has an expanding portfolio of tissues and cells for preclinical research to enable the discovery of more effective drugs. I'll provide more information on this portfolio in a moment. As I mentioned, the Life Sciences division is leveraging LifeNet Health's infrastructure, relationships, reputation, and vertical integration to provide the highest quality tissue and cells for research. We see this as a key advantage. We are leaders in this regard, and I believe the only provider that has vertical integration that enables the stewardship of the donated gift from the donor to recovery, to handling and preservation, and to the isolation of cells. Since we have control over each of these processes, we can ensure the highest quality standards are met. Our life science portfolio is growing. Here's a chart that depicts our current and future products and services. Let's start with human tissue services. This is a custom prospective tissue recovery program where we work directly with the client to define a project, identify an appropriate donor, and recover the tissue of interest for research. Human primary cells. Our initial focus here has been on liver, but will expand to other organs in the future. We have a large inventory of both healthy and diseased liver tissue products and cells, including hepatocytes, which are the topic of today's discussion. Human cell models. We have an active R&D pipeline where we're utilizing human primary cells to develop complex cellular models. For example, we're developing a fully human hepatic triculture model, as well as a human thyroid tissue model. We will discuss these in detail in upcoming webinars. Now it's my pleasure to introduce you to today's speaker, Dr. Ed LeCluyse. Ed is a principal scientist at Life Sciences at LifeNet Health and is also a faculty member at the University of North Carolina. Ed received his PhD in biochemistry at the University of Kansas, where he also did his postdoctoral training. Ed has over 25 years of experience in the field of in vitro pharmacology and toxicology, has held positions at Merck, Cells Direct, Invitrogen, and academic positions at the Hamner Institute of Health and UNC's Eshelman School of Pharmacy. More recently, his research has focused on the isolation of primary liver cells from both normal and diseased tissues and the development of novel in vitro human model systems to study mechanisms of liver disease. Dr. LeCluyse is the author of over 100 publications and has presented numerous lectures on topics such as enzyme-induced drug interactions, mechanisms of drug-induced liver injury, and in vitro hepatic culture model systems. Ed has also been a consultant and a resource for the FDA in the development of liver-focused guidance documents. Based on this background, you can see why Ed is considered by his colleagues as a key opinion leader in the field. So without any further ado, let's get our webinar started. Ed, over to you.
Thank you, Paul. Can you hear me okay?
Yes, I hear.
And will I be able to control the slides from where I am? It looks like I will be. So okay, let me start off by just welcoming everybody in attendance. I noticed a number of familiar names representing different companies, different departments, and obviously different applications, which should make today's discussion quite interesting and relevant. And I would like to mention that in particular, if there's our overall design of this particular, hang on, trying to make sure I'm able to advance the slides. There might be a delay in advancing the slides, or I can't tell whether I'm controlling the slides or not. Can somebody give me some help or guidance there? Yeah, it looks like it's I'm not able to take control of the keyboard. So if somebody wants to advance for me, that would be great. Well, So we'll try this and see how it goes. So I want to begin by just saying that this particular presentation is intended for people who are just beginning their journey with the use of hepatocytes, as well as the more experienced end users. So those that are the more experienced users, bear with me when I talk about some of the basic characterization and principles that go into basic characterization of hepatocytes and choosing hepatocytes for different applications and research areas. I'm going to then focus on more specific departmental segments, especially during drug discovery and development, where I feel there's specific needs, specific agendas, and specific questions that need to be answered when it comes to the best selection of hepatocyte lots for their particular research endeavors. And then finally, these days, with the emphasis and the emergence of so many novel culture technologies for hepatocytes in a number of different disciplines, from discovery through investigative talks, I thought that was a worthwhile topic to address and discuss characterization considerations when it comes to choosing hepatocyte lots. So can we advance to the next slide? So as I mentioned before, and many of you are familiar with the current applications of human liver tissues and cells, as well as services that are provided that are dependent on the use of liver tissues and primary cells, as it ranges from basic and translational research to, as I pointed out before, individual departments with their own agendas and focus in discovery, preclinical development, safety assessment, and even the use and application of next-gen model systems, that each and every one of these has particular agendas in mind, performance specifications that are needed or required. And what I encounter through my own interactions with colleagues and constituents in this area, as well as students that I've trained over the years, is that a simple characterization, or C of A, does not always allow you to make good decisions for your particular needs and applications. So we hope to go a little bit deeper into that in today's discussion as well. Next slide. So this is going to be a recurring theme, and one I'd like to emphasize from the beginning is, first of all, based on my own experience with working with people that are new to the hepatocyte field, especially primary hepatocytes, and whether you're a student or whether you're a first-time user but experienced in the lab, the first thing you should realize is not all hepatocyte lots are the same. So there's batch-to-batch variability. You need to be aware of the fact that there's differences based on age, or what we often refer to as life stage. Certainly, the particular donor lots that we produce are going to be range from coming from healthy individuals, as well as those with various stages of liver disease. And also there's going to be other donor demographics that need to be considered depending on your application and your particular research area, that I'll go into more detail in subsequent slides. And what we usually try to do, and what I try to focus on, is matching the best candidate batches with the particular interest of the researchers, and especially their application needs. And this can range in a number of different areas, but you'll need to start thinking about it in terms of what questions to ask and what kind of performance specifications are going to best meet your needs, such as platability, as we call it, which is really the attachment efficiency of each batch of cells, because not all batches will attach to standard plasticware, for example, or collagen-coated microtiter plates. which is probably the most commonly used platform among most hepatocyte users. But again, as I mentioned, with these advanced cell culture technologies, that it's getting more and more complicated in terms of their ability to attach to one another or engraft onto three-dimensional scaffolds or to other cell types to form three-dimensional tissue constructs or even engraft in vivo. Also, you need to ask yourself whether single-cell suspensions versus cluster or aggregate types of batches of cells are best going to serve your purpose or would be suitable for your purposes. And as I mentioned before, application and technique specific kind of questions or details are important to at least understand, but also will provide you with a little bit of guidance in terms of choosing the best hepatocytes based on what kind of research application, the techniques you're using, the platforms you might be using, and even whether you're going into 3 dimensions or not. Next slide. First, I'd like to emphasize, and that often is overlooked, is what about the role of tissue and cell sourcing, because it all kind of begins there. Paul did a nice job of sort of giving some of the importance or the benefits of vertically integrated system like we have, where you're controlling most aspects of recovery through isolation, cryopreservation, and characterization. But bear in mind is that most assays that you perform or are performed using primary hepatocytes are only as good as the cells that go into them, especially these new platforms that are being designed and developed today. So many of us historically have relied on cell lines or easy to use and reproducible type cells or cell lines, but they don't always mimic or give us the responses that we're looking for our particular research application. So it's important to understand what cell batches are going to be best for those applications, especially for those of us that are dependent on primary cells or retaining as much of that type of phenotype to model what we're trying to model under laboratory conditions. So access to quality human tissues and cells from both healthy as well as disease donors is such an important part of many aspects of what we're involved in, from toxicity testing, especially DILI and IDILI, to ADME-related questions, especially age-related and possibly even disease-related differences in drug disposition and elimination. And then in particularly for disease modeling, because you have to ask yourself, you know, is your artificial system that you devised in the laboratory conditions mimicking the phenotype of the disease in the patient population that you're targeting? And most of all, really, to be able to say that we have a human-based model system that mimics these events, it should be grounded in a good understanding of human biology and pathology, and especially to be able to answer questions about deviations or departures from what is normal. So to be able to say that something's different than a healthy individual or different from normal, you kind of have to have good definitions around that. And what are the benchmarks for that, and what are the performance specs around that? And certainly bear in mind that the inherent biological variability, which we often seek and which is often a good thing to understand and incorporate into your experimental design, it can be interfered with the artificial variability that can hamper your understanding of true biological variability. So understanding those particulars and how the different stages of hepatocyte production and characterization can impact your choices of batches of hepatocytes. And bearing in mind that the guidelines that are in place for tissue repositories and tissue banks do not often translate or are not in place for the preclinical applications. And provisional organizations. Next slide. So this is just an example of what I was just referring to in terms of like how are we going to define what is healthy versus disease versus, for example, a damaged tissue that can be due to a number of things, including warm ischemia or cold ischemia times, donor specifications, or even just improper handling or shipping practices. So especially somebody who's trying to define the difference between normal liver and disease liver by using hepatocytes from someone that at least was claimed to have a normal healthy liver from somebody who's got various stages of steatosis to NASH versus somebody who, or at least a tissue that's provided that went through some form of damage through warm or cold ischemia time. All those are going to be important issues to address and conditions to understand about the particular lots that you might have to choose from. Next slide. So it all begins with a complete donor information, beginning with the donor specifications, and that's the traditional things that are listed in the top left there, from age, gender, height, weight, BMI. All of those factors should be considerations that for your choices of hepatocytes depending on your particular research area and application. But in particular, just as important is the donor medical history. And as you can see there, it often will describe their background history and use of things like tobacco, alcohol, and drug use, for example. Serologies are mostly exclusionary criteria to make sure that you don't encounter those tissues that have been exposed to infectious diseases, especially in today's world, COVID-19. And by the way, most of the tissues that are being recovered this day and age do now include that as part of their recovery specifications, in case you're wondering or concerned about that. But also how medications and past medical history, like in this particular individual's case, that they had diabetes for 12 years or more, is going to be important for you to understand that how that might impact your particular assay requirements, but it may also be something that you're looking for if you're somebody who is studying individuals with diabetes. So next slide. For the most part, a lot of that information is also going to be included in a histopathology report that comes with most of our tissues, and I think this is being provided to a greater and greater degree, especially this day and age where there's such a focus and emphasis on fatty liver disease and NASH and related diseases like type 2 diabetes. So that comes with all the scoring system, especially a NAS score that's related to NAFLD and NASH condition of the liver. So make sure that you now understand that you can ask for that. And if you have any trouble interpreting what these values mean, especially for your research applications, don't hesitate to ask somebody that can provide you that information accurately. Most of our tissues are now examined by a liver pathologist, and then scores provided for each of our tissues. And even special comments or notes will be provided that could be of particular interest to you if you're in need of that or request that. Next slide. So some of the traditional characterization that's currently done, I think, by most providers these days, but just never hurts to review that. But some of the new considerations for that, or additional considerations when looking at some of these old ways of characterizing hepatocyte batches through different lenses, I think is a worthwhile exercise to go through. But things like the viability and yield of recovered hepatocytes, so most vials will will provide that in a C of A in terms of the viability. What you're looking for generally is batches of hepatocytes that are greater than 70%, if not higher. Depending on your application, that may be more or less important to you. And we'll get into more details about that in a minute. Oftentimes, the yield is between 5 and 10 million cells per vial, but that's based on traditional conventions that was, I think, integrated several decades ago when primary hepatocytes, or at least cryopreservation of primary hepatocytes, became fairly common and routine, and that was basically based on the assumption that somewhere between 5 and 10 million cells are going to give you a single microtiter plate's worth of experimental assays. However, in today's world, sometimes people require more cells per vial or would like to have more cells, and some less than that, especially with microchip technologies and things like that. So just make sure that you're asking those kind of questions in terms of those, especially that require more or less cells per vial for your given application. In the morphological assessment, again, it's generally qualitative, but it can be very helpful in choosing your batches of cells, especially for toxin disease applications. The next category down is what we refer to as plateability. That has mostly to do with their attachment efficiency for collagen-coated plates. Typically, these are 24-well format plates that's used as sort of the standard for determining attachment efficiency. And it generally is used to dictate whether a cell is going to be designated as a suspension lot or a platable lot. But there's also the different categories of plated cells in the sense of their monolayer stability, how confluent the monolayers are, the longevity of how long they will last in culture, and also the integrity and maintenance of normal morphological features, and also hepatic functions over time. In particular, I want to mention that now with the growing interest in hepatocytes from disease donors, that a lot of these can be different. A lot of traditional culture platforms will not house hepatocytes from disease donors as effectively as a traditional healthy hepatocytes. So bear in mind that you may need to ask, or you may have to consider different platforms that I'll get into in a little bit later, especially if your focus area is in disease versus healthy hepatocyte comparisons. And again, the histopathology is becoming more and more important, but make sure that you're getting that or you're requesting that, especially as it may apply to almost every application. These days, there's noted differences in healthy versus diseased liver tissues, whether you're looking at drug metabolism and elimination, or whether you're doing hepatotoxicity testing or disease modeling. Next slide. So some of the other additional traditional data that's provided, enzyme profiling of most lots. This goes back to the traditional preclinical drug development and FDA and EMA requirements or recommendations in terms of the use of hepatocytes for drug metabolism studies as well as drug-drug interaction assessment. So most of the major CYPs, as well as some of the UGTs or a generic marker like 7EC, information about those values are provided. If you're not familiar with that, make sure that you're asking about the enzyme profiling data and whether it represents normal values or average values, depending on whether you're talking about an individual donor or pooled lots of hepatocytes. For the DDI or induction potential assessment, this has evolved over the past 5 to 10 years. Currently, the state of the art was recommending that RNA be focused on both the FDA and the EMA guidances, now mostly focused on RNA induction or changes relative to a positive control. And the latest recommendations by the induction working group from Big Pharma is that about a 10-fold induction with a positive control like rifampin be considered as a gold standard, or at least a new standard, as compared to traditionally it will range from about 5-fold to about 10-fold. And then also bear in mind that depending on your applications, other optional or even custom type characterization may be beneficial if not required, especially for transporter expression or functional activity, for looking at biliary excretion or even cholestatic events, as well as other pathways of elimination or oxidation, as well as reduction events. And many of these are fit for purpose, which I'll get into more in some of the specific subsegments of applications and qualifications. Next slide. So one of the other things that I'd like to focus on, again, is the biological variability that is often valued and a benefit to understand what the inter-individual variability might be, or even age-related differences between healthy and disease, can often be challenged by the unknown and ignored technical variability. So one of the things that groups are working towards, and especially within our organization, is better standardization of those methods from recovery through isolation, cryopreservation, and even characterization. Part of that is just getting technical consistency, making sure we're using best practices for organ handling, flushing protocols when it comes to making sure that the organ's well preserved and well handled during the time of recovery on its way to the laboratory to be processed. So, and then just the years of in-house experience or expertise that we have using standardized processing for isolation and cryopreservation. But one of the things that you have to be mindful of these days is understanding donor variation. And when I say donor variation in this case, I mean of the tissues during recovery, that each of those need to be considered differently, and depending on whether they represent a healthy phenotype versus a disease phenotype, that the protocol needs to be adjusted accordingly. So make sure you're asking, or at least you're being mindful of the practices that are being utilized or the protocols that are being utilized for the isolation and processing of hepatocytes, especially from diseased donor tissues. Because if you use the same methodology for everything from a healthy to a diseased tissue, then you're going to change the phenotype of those cells that are collected by the end of the processing. And that may or may not be of relevance to you, but it's certainly worthwhile being mindful of that. Next slide. So I'm going to go into some individual application-specific characterization strategies, and this is partly based on my own experience or my own conversations with colleagues over the years, but also recognizing that we need to raise the bar, we need to sort of up our characterization process to better meet the needs and requirements of these different groups. So we're going to jump in and start with the discovery group. Next slide. So when I think of the different kinds of considerations that go into anybody's decision, these are often the common topics that come up, or questions around these topics. that you should be mindful of, or they're definitely worth making note of and making sure that you're thinking about these things when you're assessing or trying to qualify different batches of hepatocytes. Many of these we've already discussed, and just trying to reemphasize is the degree of platability. And remember, this has to do with the attachment efficiency of the cells, how effectively they work in different plate configurations, but also different platforms. So it's gotten a little more complicated over the years. And bear in mind that something that attaches just fine in a 24-well plate or a 6-well plate may not necessarily work well or attach effectively in a 96-well or a 384-well format. Again, as I mentioned before, bear in mind that life stage can be important for you. Now, I have a number of colleagues that life stage is important. They actually want to study differences in metabolism or toxicity in different life stages. So bear in mind that there's developmental issues, especially in neonates up to about 2-year-olds, where things begin to then reflect more of the adult type phenotype. But we get asked all the time, you know, are kids that different than adults? And for instance, are teenagers that different than adults? And where do I draw the line? And typically it's arbitrary and artificial rather than really based in developmental human biology. So again, depending on your needs and your applications, be mindful of those things. And of course, the bigger consideration these days in particular is the difference between hepatocytes from a healthy liver versus a diseased liver, and making sure that you're getting proper qualification, especially in the area of disease modeling and in toxicology. And also the number of cells per vial, as I implied before. Fewer cells per vial are sometimes in higher demand for things like microchip technologies or some of these new fluidic technologies because they require fewer cells per chamber, for example. Some people need a greater number of cells depending on the number of experiments that they're running, et cetera. So there's not necessarily anything magic about the number of cells per vial, but just make sure you're being mindful of that. Percent viability is always a big one. Generally, our vials are intended to deliver something more on the order of like 80% plus viability post-thaw, but bear in mind that your requirements may be less stringent or more stringent than that, especially if you're doing very discriminating, discerning techniques like high content imaging, for example. And we'll get to that a little bit later as well. And also, bear in mind that not all lots are of equal size, so the number of vials per lot may be important for you, depending on whether your needs require only like 10 vials or sometimes 1,000 vials. Next slide. So this next segment, I'm going to break these up into a focus on basically clinical or preclinical development from discovery through development and safety assessment, and try to highlight as best as I can from my own experience in conversations with colleagues about some of the more important considerations, and helping provide you some better guidance or more guidance than what you might traditionally get. So next slide. So starting with the early stages of development and drug discovery, especially where, as the cartoon indicates on the right-hand side, where you're trying to retain or maintain the phenotype, especially from a disease donor or disease tissue, and translating that to in vitro model. So one of the things that often comes up in discussion, should I take hepatocytes from a normal healthy liver and then try to induce that phenotype under laboratory conditions? Or should I just take hepatocytes from a diseased tissue and try to maintain or retain that? Now, I think they're both legitimate approaches as long as you know what questions you need to answer, and be aware that they do arrive at different points. So if you artificially induce a disease state into your hepatocytes from a normal, healthy liver, at least gene expression data that's out there in the literature would suggest that you can't recapitulate the exact phenotype, the gene expression profiles, biomarker, and pathway criteria of those cells from actually the cells that come from a diseased tissue itself. So some of the other considerations are listed there on the left about platability and longevity. So there's no magic to that. Many of my colleagues are doing experiments over a 24 to 72-hour time frame, so short-term platable is often enough, or even suspension cultures are often sufficient. But others, they require long-term cultures. They want to do them in 96-well plates or 384-well plates, and they want or need the hepatocytes to last for 2 to 3 weeks in some cases. So bear in mind that not all hepatocyte batches that may meet your disease criteria or your healthy criteria, they may not actually meet your performance specifications. So it's sort of a 2 or 3 pronged set of questions for characterization or identifying the right batches of hepatocytes, and including looking at, again, at the histopathology reports and other considerations. Next slide. So again, depending on what area in particular you're focusing on, from disease modeling and an advanced or next-gen culture platform, or doing pathway analysis, target ID, or even efficacy screening, to make sure that the specifications are fit for your purposes, especially making sure that certain pathways and biomarkers are retained. I think that's one of the biggest challenges these days, is making sure that, especially if you're going to transfer cells from a vial into a particular culture platform, that they retain the original phenotype and pathways and other biomarkers of importance or relevance to you is one of the biggest challenges. Next slide. So we've begun to put together sort of a little guidance table for each of these different niche areas of research, with their own unique qualifiers, if you will, or specifications that are needed. So you'll see this throughout the different segments, and we can provide these for you if you find these useful. But without belaboring each of the individual rows or individual types of applications or types of experiments, just want to point out that these are each of these columns represents a different characteristic, if you will, or attribute that may or may not be important for your particular type of studies. And so everything from the donor specs and medical history to attachment index, which relates to the attachment efficiency, metabolic capacity, cell polarity and architecture, and whether you need to retain that, whether a monoculture or multicellular culture co-culture system is important, is considered here, and 3D index refers to their ability to form three-dimensional aggregates, whether it's spheroid culture or form three-dimensional tissue constructs, or even engraft in vivo or in a scaffold type situation is considered here, as well as what we call a purity index, which is making sure that the composition or the ratio of hepatocytes to other contaminating cell types meets your needs and qualifications. Next slide. So moving on to the DMPK applications and studies, these are more the traditional ones, so I'll skim over most of the basic principles here, but again, in consideration of maybe some of the people new to hepatocytes, it is important for you to understand what your particular attributes of your cells might be or might need to be for these preclinical ADME assessments. Most of this is going to be based on the US FDA and EMA DDI guidance documents, where the focus is going to be on the ADME pathways, sometimes on transporter expression and functionality, but also other key pathways, FMOs and aldehyde oxidases, and sometimes even reduction pathways and other phase 2 metabolic pathways may be of importance to you. So make sure that you're looking at that or examining that in the C of A's. So next slide. One of the things that I often feel or always feel compelled to remind people of is that the heterogeneity at the microarchitecture level of individual liver tissues. And where that comes into play is especially if you're doing drug metabolism, DDI kinds of assessments, but also this will overlap with looking at toxic events in the liver. But if you look at the histology slides in the lower left-hand panel and the middle panel biopsy sample that are stained with antibodies to two majorly expressed proteins in human liver, namely 3A4 in the lower left-hand panel and OATP1B1 in the middle biopsy segment, notice the immunostaining pattern is not ubiquitous throughout the liver tissue, and this is common in almost all adult liver tissues for major CYPs as well as major uptake and sometimes efflux transporters. So what's interesting, if you really think about it, when we isolate hepatocytes from healthy adult livers, you're going to get a mixture of phenotypes, especially zone 1 and zone 3 type phenotypes is represented in the upper right-hand corner in the in vitro cultures. So these are control and RIF-treated cultures on the right-hand column. And these are actually confluent monolayers. So the dark areas that you see that look like they're devoid of hepatocytes actually do have cells there. They just don't stain with antibodies to 3A4, reflecting that heterogeneity that's actually expressed or exhibited in whole liver on the lower left-hand side. And the same thing for transporters, bear in mind that heterogeneity is exhibited. Now, curiously, if you treat them with an inducer of 3A4, notice that those cells that were formerly dark will now light up, so suggesting they are competent in responding to PXR agonists, but also in expressing the gene, they just don't. And that's one of the other things that you have to bear in mind, especially in life stage related experiments, as well as at different age groups, that the differences in the expression level can vary, as well as things like ploidy, whether they're small diploid hepatocytes or whether they're larger polyploid hepatocytes. The gene expression profiles will change with that. So each batch of hepatocytes varies slightly depending on the donor with these kind of patterns, by the way, and also what drugs or medications the donor was on prior to the isolation of the cells. Next slide. So hepatocyte users in the drug testing program, especially for DMPK applications, your needs are going to be based on those kind of considerations and the US FDA and EMA guidance documents. You typically going to use suspension hepatocytes for doing basic metabolism, although these days platable hepatocytes are being used for a number of applications, which I'll get into in a second. So things like platability, whether they can be accommodated in 96 well or 384 well platforms. Metabolic capacity becomes more critical than maybe other types of applications. You'll want to look for things like whether they're qualified to meet the minimum specs as recommended by the induction working group for example, that published a recent paper in DMD, by the way, and other considerations that may be relevant to your particular area of study or your particular therapeutic area. Next slide. So in particular, enzyme profiling and of all the lots becomes a slightly different reflective exercise if you think about the difference between healthy and donor or healthy and diseased donors. It's pretty evident nowadays that differences in cytochrome P450 and especially nuclear receptor biology and transporter expression levels are significantly different in hepatocytes from healthy individuals versus diseased individuals, especially with NAFLD and NASH. One of the other considerations is the short-term metabolic clearance assays, especially for the low turnover compounds that now we're starting to get more and more questions about whether our hepatocytes are able to discern turnover of highly stable compounds or low turnover compounds, as they're sometimes called, to get good pharmacokinetic information for those particular compounds. And then, of course, if you are looking for transporter interactions, making sure your batches of hepatocytes exhibit good uptake and efflux capacity, especially in a vertically integrated fashion from uptake through efflux for biliary excretion purposes. Next slide. And this is the corresponding table, something to consider. It's sort of as a guidance if you're working in DMPK applications, the importance of these different relevant attributes, if you will, or features that might be of consideration for DMPK types of functions and activities. Next slide. So the next segment I'm going to focus on is the in vitro toxicology, or whether you're in safety assessment or investigative toxicology areas. Just again to reiterate, if you're new to this area, that there continues to be unexpected and unexplained hepatotoxic events in early clinical trials that were not identified through preclinical development, especially in animal models, suggesting that we still need to be doing a better job of assessing that preclinically, but also mostly that we need better tools and better model systems, all of which begins with the cells that you utilize for these kind of applications, as well as the platforms and how elaborate they are. Next slide. So in order to mimic the progression of often toxic or pathological responses in events, there's a lot more challenges in selecting and picking batches of hepatocytes, especially when you're trying to model complex diseases and toxic events that are progressive over time. And that's been at least one of the biggest hurdles, at least when my discussions with other colleagues in the investigative tox programs, but also in the discovery programs. Because these events often require, multiple cell types in a 3D configuration or architecture to really mimic those specific types of events. So you now are dealing with whole different requirements or specifications for your cells, as well as those that work well in your platforms, these new technologies. And then also they may require longer periods of time to exhibit these kind of details, let alone be able to study them. And sometimes even donor match cells may be a prerequisite or requirement to identify particular events, as well as pathways or biomarkers to focus on and put your attention on. And it's become more and more apparent that the role of inflammation and adaptive immune responses are often that are traditionally been overlooked, but certainly not provided in just monocultures of hepatocytes by themselves. Next slide. So for these particular applications, human hepatocyte needs can vary from the other research groups. There's going to be some overlap in terms of platability and monolayer integrity. In particular, bear in mind that you need to know where your hepatocytes are starting from, especially on the stress scale of things, whether that's they've experienced some oxidative stress either prior to or during isolation. You're going to need to know that and be able to have a benchmark of what you're going to consider normal or departures from normal. You need to know where your cells are starting on that scale. So metabolic capacity is also important to mimic some events, including reactive metabolite formation, or which could become an issue with some of your toxic events. Histopathology, as I was alluding to, is an important consideration. And bear in mind that phenotype from hepatocytes from a healthy donor versus a diseased donor is going to be different, and therefore change and affect your responses. Cell polarity, especially if you're looking at cholestatic events, is going to be key in having hepatocytes that meet those specs, or at least are amenable to those, especially for 3D formation and engraftment type studies to scaffolds or formation of three-dimensional tissues. Becomes additional attributes and considerations for DILI and iDILI in particular. Next slide. So again, to reiterate or emphasize a fit for purpose specifications of hepatocytes for investigative tox applications, whether it's for the potential for DILI, the best model system, whether 2D or 3D meets your needs, monocultures versus co-cultures. All of these are considerations for getting proper inputs for computer modeling, for high content imaging, as well as mitotoxicity, which are all key types of applications and assays to run that you need to be aware of your hepatocyte batches and their particular characteristics that best meet your needs. Next slide. So this particular table, again, just what's interesting, well, I guess because I'm biased, since I've worked in the hepatotoxicity area for as long as I have now, and my appreciation for how most, if not all, these attributes are key inputs or key considerations for picking the proper batches of cells for my applications and knowing where you start. This table at least gives you some guidance and for questioning, asking questions about different batches of hepatocytes, but also consideration for whether they're important for your needs or not. Next slide. So the last segment of my presentation, I just feel, because this conversation comes up over and over again in my discussions with colleagues and customers, collaborators, this current trend towards more 3D organotypic model, like what exactly does that mean? What are the different qualifications or characteristics of hepatocytes do we need to consider? It's worth a discussion, in my opinion. So next slide. So one of the things that we're finding ourselves in the position we're in these days is that we've reached this crossroad where clearly there's a growing demand for human-based model systems, especially to replace animal models as much as possible in disease modeling and trying to recapitulate or understand toxic events. You know, animal models typically don't work. Conventional cell culture models often don't work or give us answers to the more elaborate or detailed questions we need. And there's therefore an extensive effort has gone into forming multidimensional and multicellular in vitro culture systems that the expectation or the desire is to get them to recapitulate a lot of these events, which is a challenge in and of itself. And there's a number of 3D organotypic culture platforms that are out there. Many of you are using those or testing those, evaluating those. And of course, a lot of their reports is that they recapitulate and sustain dynamic cellular events observed in vivo, but what are their benchmarks and what particular cell types did they use to validate their model systems? You can appreciate the need for more standardization in this area, but also probably some kind of consensus on what the proper validation ought to be. Next slide. So by my way of understanding, or at least of liver biology and pathology, toxicology, that some of the important characteristics that need to be considered, and at least you need to ask yourself, is the importance of retaining that zonal architecture and histotypic characteristics that I described before, including the characteristic differences in gene profiling or gene expression across different zones. Because sometimes that relates to hepatotoxic events, if not the progression of diseases like hepatitis and NAFLD and NASH, based on the mechanism of action. So the relationship between now hepatocytes and the other cell types becomes more critical and important, especially in terms of their distribution and proportions. So if you're going to use a co-culture model, ensuring that their distribution and ratio, if you will, or proportions to one another is in keeping with modeling a healthy condition or a disease condition. You need to be mindful of those things because that's going to impact their molecular and biochemical pathways that are expressed, the gene expression, nuclear receptor activation pathways. Also, the longevity and the metabolic capacity of the model system is going to be impacted by both the platform, the overall architecture, the cell types that are involved, as well as the robustness and reproducibility of the model system. So all these now need to be considered with these more elaborate, complicated model systems. And then accessibility just refers to the fact that many of these platforms, we still want to have access to them and be able to poke them and prod them and study our normal